So I'm Bob Schwartz. I'm a Cohen Distinguished Professor in pharmacobiology and biochemistry. And my research is basically the topic that's going to be discussed today. It's on regeneration of the failing heart or damaged heart. I also have an appointment with the Texas Heart Institute where I'm a director of a stem cell laboratory. So I know Mark Mercola over many, many years, at least 15 years. And most excitingly, in the last 3 years, we began to collaborate. And Mark's studies are unique for the breadth and, I would say, brilliance that they bring into it. He's one of the first people to use biological chemistry, in a sense, to find and screen for new drugs, molecules that regulates developmental processes. And he'll talk a little bit about that. He's also, by doing these screens, he's discovered new pathways, new signaling pathways, new gene targets that are involved with the process of cardiac regeneration, starting at the level of an embryonic stem cell. He's supported by many NIH grants and also by support from the California Stem Cell Institute, which he was one of the original scientists that got funded through their activities. Mark is a leader at many levels, as I said, developmental biology, and at the level of cell signaling. And it's a real pleasure for me to have Mark here. You can read all the information about him. He's a director at Sanford Burnham, and he's done lots of amazing things. But to me, he's one of my best friends, and I really enjoy listening to Mark talk because he gives a great talk.
Thank you very much. Well, Bob, thank you very much for the invitation and the wonderful introduction. It's truly an honor to be introduced by you. I consider you a tremendous resource for the field. Respect you and have admired your work. As I said, it's wonderful to be here. Thank you very much. I'm going to tell you about our work to try to regenerate the heart. I'd love to be able to stand here and tell you we've got miracle drugs that will do that, but that's not the case today. But what I'm going to lay out for you is a roadmap. Microphone is not on. What do I need to do?
You want to use the podium one?
I can use the podium one, that's fine. Sure.
Yeah, that one's picking up pretty
Okay, very good. So I can hear it now. So what I'm going to tell you is basically a roadmap or a strategy to come up with drugs that I think will regenerate the heart. And I think the importance of doing this in academic centers is that this work is fairly basic. And what we'll try to do is identify the drug targets that then will be taken forward, hopefully, by pharmaceutical companies to produce the molecules that will eventually be introduced into the clinic. So I don't think we really have to belabor the point about the damage that a heart attack causes. After myocardial infarction, you can lose upwards of 1 billion cells, typically in the left ventricle of the heart. And the heart's fundamental pumping mechanism is seriously compromised. And so in its essence, basically, the loss of muscle cells needs to be replaced. So a question then is how to go about doing that. The current therapies do not address the loss of muscle cells, but rather try to unload the burden of the heart. And so what I would like to do is develop a way that we could discover the drug targets that will be used to find drugs to cure the heart. So the major problem is that the mammalian heart does not regenerate itself. So this is in stark contrast to the hearts of lower vertebrates. They can regenerate themselves scarlessly. And so regeneration is something that people have known about for many, many years. And I'm going to show you a video that was made a number of years ago showing the regeneration of the limb of a newt. Now, newts, okay, I think you saw it in the real life, and now you're going to see it schematically. And it introduces a couple of key points. When the limb is cut off, what forms is a blastema over the surface of the wound. And this blastema consists of cells that have de-differentiated. They're cells that have lost their phenotype. So bone cells, nerve cells, muscle cells have lost their characteristics, their differentiated characteristics, and have formed, in essence, a covering of stem cells over the wound surface. And with time, these cells will proliferate and lay down new structures. This has been something that people have known about for many, many years, but are only now beginning to understand the process. So some of the themes that I'm going to talk about are: where do these cells come from that can regenerate? Is limb regeneration similar to regeneration in the heart? And if we can identify cells within the heart that are the basis for regeneration, can we find a way, a pharmacological way, to increase their number or increase their ability to give rise to new heart muscle? Okay. As I said, the hearts of lower vertebrates can also regenerate beautifully. And here's an example of the scarless regeneration that you see in a fish heart. If you take a fish heart, and you basically, it's diagrammed here, you just go in there and you cut off the bottom of the heart, the apex of the heart, throw that bit away. The heart will, and you can see it will first begin to form, in essence, a little bit of a scar in the wounded area, but then that will be replaced by new heart muscle cells. Okay? And this is something that our heart just simply cannot do, at least cannot do very efficiently. For many years, it's been quite controversial about whether the mammalian heart, in particular the human heart, can regenerate at all. And in fact, much work during the 1960s, 70s, and 80s argued that the heart just simply is non-regenerative. And we'll go through I'll show you some of the data that was used to make that case. But over the last 30 years or so, this view has gradually given way to the view that the heart can regenerate itself, albeit very, very little, very inefficiently, and certainly not well enough to overcome the massive loss of heart muscle cells that occurs after myocardial infarction or following other heart disease. Part of the problem and part of the debate and the controversy, especially at meetings, I mean, there have been screaming matches at meetings over the question of whether or not human heart muscle can regenerate. Part of the controversy is that depending on the techniques you use to try to answer the question, have given rise to different results. So in some cases, people will say, no, we never ever see a replicating cell. And in other cases, people have said, well, every year, about 30% or so of the muscle cells can be replaced so that the heart can completely replace itself every 8 years or so. So what is the reason for the discrepancy? So here's a figure from a landmark paper from 1925. In this paper, and I don't have my glasses on, so I'm going to have a hard time looking at it. In this paper here, what they're scoring is the number of nuclei per field, the nuclei of individual cells on histological sections of a heart. And let's see if I can get this. You get a pointer here that stays on. Hang on. Okay. So if you look at these data here, this is a histogram of frequency. So here we have the number of cells on the x-axis. The number of nuclei on the x-axis and the number of cells with that particular number of nuclei on the y-axis. And it's too small for you to read, so I'll read it for you. In the normal heart, most sections or fields of view have 10 nuclei. And in the hypertrophic heart, it's the same. And if you look at the atrophic heart, it's about twice as many. So the conclusion from this landmark paper is that there aren't any more nuclei in the hypertrophic heart. So the heart that enlarges in response to stress enlarges because the individual cells get bigger, not because you grow more cells. That's a very important conclusion. Okay? It doesn't say, however, that there is no regeneration. In the atrophic heart, there are more nuclei. That also doesn't say there's any regeneration because the additional nuclei could be due to other cells in the heart, such as fibroblasts that aren't heart muscle cells. So these experiments simply don't get at the question. However, these experiments are the root of the debate about whether the heart can regenerate new heart muscle cells or not. And I'd like to digress a little bit just to show in cardiology how long an issue that is considered dogma can persist. So this is a diagram that summarizes what was thought from Galen of Pergamon, based on Galen of Pergamon's explanation for how the heart pumps blood and how blood circulates in the body. And Galen felt, and this is many of you are probably well aware of this, Galen felt blood was generated in the liver and pumped through the heart to the muscles where it became muscle cells. Okay? So it was a one-way flow. Of course, this would take a huge amount of blood and a tremendous amount of energy to generate that blood. But this took 1,500 years to dispel this argument. And shown here, I pulled this off the internet the other day, is an interesting manuscript. This is a treatise by Galen that had been copied laboriously for 1,000 years. Okay? And it was copied in Greek, and you can even see the Latin commentary on the side. So this propagated this view that the blood just originates in the liver and flows one way. And it took 1,500 years to begin to chip away at this concept. And so the early anatomists, shown here Leonardo with drawings of the heart, identified a venous system. And of course the venous system was well described by Vesalius. And so this slowly gave rise to the underlying, or gave people impetus to question this philosophy. And finally it was William Harvey who through direct experimentation showed that blood must flow back to the heart. In other words, the blood circulates. So 1,500 years to dispel this idea. So does the human heart regenerate? So there, of course, there is a counter view that the heart is capable of some regeneration. People have been noting incidents of increased numbers of nuclei and indeed dividing cells in heart muscle cells for many years. But a question has been, is this very frequent? Does it really exist? Are they mistaking other cells in the heart for heart muscle cells? And I'll show you one picture of what was the opposing camp. That's heart muscle cells do indeed get turnover and get replaced. This doesn't show that heart cardiomyocytes are dividing, but what it does show is that there is a stem cell population, or purports to show, a stem cell population that can replace the cardiomyocytes. So in this figure, what you can see here in blue are the nuclei of very small cells, the cytoplasm of which is identified in green, and those cells are containing speckles within their nuclei that are a transcription factor that is used to identify those cells as cardiomyocyte progenitors. So the argument from this is that progenitor cells, not cardiomyocytes yet, but progenitors to cardiomyocytes exist in the heart, perhaps set aside during development. They replicate upon injury, they differentiate, become new cardiomyocytes, and can rebuild the damaged heart. So this was the opposing view to the idea that the heart muscle cells never turn over, that the cells that you're born with are the cells that you die with, at least hopefully that you die with them. So how to resolve this controversy? So everyone here who was, well, those of us who were alive before 1963, when above-ground atomic testing ceased, are all basically subjects, in essence, in a global Pulsechase experiment. We're all labeled with C14 due to the above-ground testing of atomic weapons begun in 1945 and terminating in 1963. So during that period, when C14 was released into the atmosphere, it would label anything living, anything that would take it up. And so using this, that's the pulse, 1945 to 1963. So all of us were labeled during that period. And then, of course, after 1963 to the present can be viewed as the chase, what we scientifically call the chase. At that point, you're taking up not C14, but you're taking up normal carbon-12. And so you can ask then, if a cell that was in existence in your body during the pulse is still there, or has it been replaced? In other words, has it been diluted in your body? And by doing that, there was a very nice paper in 2009 by Jonas Friesen and Stefan Jovinje, and this paper showed that cardiomyocytes turn over at an annual rate of 1% per year for a young adult, 25, slowing with time, with age, but nonetheless, 1% per year. That's a dramatic number. It's not the 20 or 30, 40% that had been argued before, but it's certainly not zero. And the cumulative effect could be quite meaningful. So there have been other estimates since then, and they tend to hover at about that same incidence, about 1% per year. Now, if the cells are being turned over, where do new cardiomyocytes, new muscle cells, come from? So, in principle, there are three possibilities. So if you've got a heart, or we do have a heart, in your heart, and if the cells are turning over, you could have three sources for these new cardiomyocytes. These muscle cells could come from pre-existing muscle that enters into cell cycle and divides. Just like when you cut your skin, you cut through the epidermis into the dermis, you'll see a proliferation of the cells in the area. And so the new tissue that you've created, or that is created, arose from cells that had been there already. So pre-existing cardiomyocytes could re-enter the cell cycle and divide. Alternatively, you might have some sort of professional or resident stem or progenitor cell in the heart that's been induced to proliferate and give rise to new heart muscle cells. And you might even have some cells that come from somewhere else, some other niche in the body, perhaps the bone marrow. And those cells could enter into the heart, be recruited there by cytokines, brought into the region of damage, and try to rebuild it. So to discriminate between where the cells come from, people have been using a technique called lineage labeling. And by one way or another then, they label the cells, each of these populations, the pre-existing myocytes, the endogenous stem cells, those are rather hard to label, or the bone marrow cells, and they ask, do the new heart muscle cells come from one of these sources or another? Okay? So if we go back to the fish and its beautiful scarless regeneration, what you see here is that all the pre-existing myocytes, cardiomyocytes, heart muscle cells, were labeled with a green fluorescent protein. You can easily spot them. And so then the question is, when you whack off the tip of the heart and then it can rebuild itself, will those new cells be green or will they not be green? If they're green, they had to have come from the pre-existing myocytes. If they're not green, they came from one of those other sources of cells. Okay? And the answer, shown schematically here, is that they come from the pre-existing muscle cells. So I'll go through this. Here's a diagram of the cells in the heart, just basically cells in the apex of the heart. And they're not labeled. You come along and this is a genetic trick where you turn on the expression of this green fluorescent protein only in the heart muscle cells. Bob is a real expert at this type of technology. You then have labeled all of the cells green. What's not shown here is the amputation where you remove some of those cells. But then you come back and you look several weeks later at the heart and you find that the new cells are green, meaning that they arose from the pre-existing cells. And what you see here in these real photomicrographs is the label for the new cells in the nuclei, and you can see the cardiac muscle cell with its striations is clearly green. And they go through a great deal of effort to prove this point. So if you do that in a mammal, an adult mouse, you get the opposite answer. So this was an experiment done by actually a good friend of mine, Richard Lee, in Boston. So what Rich did is he did the exact same trick, and he labeled, it didn't quite work as well as in the fish, but most of the cells in the heart green. And then instead of just going in there with scissors and cutting off part of the heart, what he did is he created a myocardial infarction by tying off the left anterior descending coronary artery. So that basically gives the animal a heart attack. Because of the lack of oxygen delivered to the tissue distal to the ligation, those cells start to die. And then he asks whether any new cells, there isn't a lot of replacement distal, just right in the infarct region, but in the area right around it at the border, there is some replacement going on, and he asks, where do those cells come from? And what happens in the region around the infarction is that there's a dilution of the green labeled cells, the proportion of cells that are green. So they went from 80% to 65%. So that would indicate that the new muscle cells came from some other source, a progenitor cell, not from a cardiomyocyte, but from something else. Okay, so to summarize what I've told you then, heart regeneration in fish and mammals differs. And it differs importantly by this, not just in the quality or the robustness of the regeneration, but importantly, the source of the cells that give rise to the new muscle cell. In the mouse, the modest regeneration seems predominantly due to a stem cell population that gives rise to the new muscle cells. Okay? The technique that Rich Lee did, the genetic labeling, didn't even have the sensitivity to pick up the replication of pre-existing muscle cells. Now in the fish, on the other hand, the pre-existing muscle cells do regenerate, are the source of the regenerating cells. So is the mouse or mammals, are we completely devoid of the ability or completely lack the ability for our muscle cells to regenerate through replication? And the answer is no. At birth, and here's a neonatal mouse pups, at birth we behave pretty much like the fish. You get very nice regeneration if you were to go in and surgically or remove part of the heart, or you were to damage the heart. And furthermore, what's really interesting is that in an adult heart, if you introduce one of a few molecules, or you genetically modify the heart in certain ways to override the terminal differentiation, the quiescence of the cell, you can trick that cell into re-entering cell cycle and dividing. So an adult heart muscle cell, albeit very, very rarely, can be tricked to re-enter cell cycle and divide. Here's an example of that. And the three types of molecules that I'm aware of that have been used for this are fibroblast growth factor, neuregulin, and periostin. Okay? So these molecules, when delivered to the heart, can stimulate some level of regeneration. Very infrequent, but some level. And in fact, they all have, in the mouse anyway, for short-term protective effects. So if the heart muscle can re-enter the cell cycle, what then keeps it from doing so? And that's a major, major question. It's not so easy clinically. I should point out, as I said, that there are adverse effects. There are clinical trials where people have tried to give that fibroblast growth factor for other reasons, largely for regenerating vasculature in the heart. But those ultimately led to problems. So you can't just try to treat a heart attack by giving some of these factors. It doesn't necessarily say that making your heart muscle cells replicate is bad, but clearly the factors can do multiple things. Okay, so what's the prospect then for regenerating for the heart, in particular a drug-based form of regeneration? So to summarize then, they would like to target a stem cell population, because that seems to be the predominant way that mammalian cells regenerate. But we could also consider targeting the pre-existing muscle cells and coaxing them to re-enter cell cycle. So basically, is it possible to model this? So how do we discover a drug that will do it? We have to in some way come up with a model system, because people have had a hard time putting their finger on exactly what is an adult cardiac stem or progenitor cell, and certainly getting them in large enough numbers to put in high throughput drug discovery screens like I'll tell you about is right now hard to even imagine. So we need a way to model regeneration in a dish robustly and cheaply so we can look for drugs that will stimulate this. So I've always been enamored with Nicholas Culpeper, who claimed that it is possible, who was one of the people who argued years ago that it is possible to find drugs to treat almost any disease that you can imagine. And here is some text that came from a treatise of his. It was actually, I think, the front page. And he argued that you should consider by what planet, in other words, what problem, what planet the afflicted part of the body is governed. So what it actually is the mechanistic basis for the problem. And then you may oppose diseases by herbs of the planet opposite to, that causes them, as diseases of Jupiter by herbs of Mercury. So you would provide something that opposes. We're not talking about homeopathy, but we're talking about actually finding a drug that would treat the root cause of the problem. So you would be able then to identify the problem mechanistically. I interpret this as identifying the problem mechanistically, modeling it, and coming after a drug, and coming after it with a drug. So how might we do this? What are the potential ways that you could boost heart regeneration? Well, you could promote new muscle cells, as I was saying, by replicating, by promoting the proliferation and differentiation of the progenitor cell. You could provide new cells from outside. I'm not going to talk about that. Transplantation, that's a huge area. You could also try to protect the heart from problems in the first place, so you protect those cells from dying. All of this could be used to regenerate or protect the heart. So what I've spent many years doing is trying to model regeneration with progenitors that are not derived from the heart, but are derived from embryonic stem cells. And the reason for this is, as I said before, it's very difficult even today to imagine getting enough adult cardiac stem or progenitor cells in a dish to use in high throughput biology. So what we've been doing is taking advantage of embryonic stem cells, which can generate basically every cell type in your body. And so I think all of you are familiar with embryonic stem cells, so I've only put one slide here to illustrate that they come from the inner cell mass of a pre-implantation embryo. They've been coaxed to grow in culture. They can be passaged indefinitely. And those cells, upon appropriate conditions, can give rise to every single cell type in the body. In fact, in a mouse, where we can easily do the experiment, if you put the embryonic stem cell back into a pre-implantation embryo, those embryonic stem cells will indeed give rise to all the cells in that mouse. And that's very clear. For instance, if you take ES, embryonic stem or ES cells from a black mouse, and you were to put them into the blastocyst of a mouse with white fur, and you would now see, in essence, a mosaic mouse. Okay? So and eventually that mosaic mouse, two of those bred to each other, can produce then the mouse of the same genetic makeup as the cells that you had in the dish. It's truly remarkable. You can take an embryonic stem cell, create any cell type you want. Human embryonic stem cells exist, and more importantly now, there's the ability to create, for all intents and purposes, an embryonic stem cell derived from cells in our own bodies. We can take our skin or other cell types and revert them back to an embryonic stem cell. So you have, in essence, stem cells, pluripotent or totipotent stem cells derived from you, meaning if you carry a genetic disease, so will those cells, and you can make, say, heart muscle cells from them, and those heart muscle cells will have that disease. This is a tremendously powerful technology. So here is a colony of embryonic stem cells, and after appropriate differentiation, you can make beating cardiomyocytes here. And these are cells that we've engineered to fluoresce red when they become cardiomyocytes. This, in fact, is our most used screen line of human embryonic stem cells that I'm going to describe to you in a number of experiments. So, using embryonic stem cells, my lab and many others, and using embryos even before that, have plotted the discrete steps that it takes to go from an embryonic stem cell or an inner cell mast cell of an embryo through making all the different cell fate choices that you need to make to come out at the other end as a cardiomyocyte. And so you first have to make, the cells have to make the different germ layers, ectoderm, mesoderm, endoderm in the embryo, ectoderm being all of our skin and nervous tissue, endoderm being the lining of the gut basically, mesoderm being everything pretty much in the middle. So once you make that mesoderm, you have to restrict it to a cardiac or cardiovascular fate, and from within a cardiovascular fate, you can make heart muscle cells. So the endogenous cardiac cells, although there's controversy about exactly what they are, how many there are, where they might be in the heart, when people isolate them and characterize the genes that they express, the molecular markers, they seem to have some overlap with the progenitors that come up developmentally. So this suggests to me that if we had drugs that would enhance the replication or the proliferation of these different stages, going from this step to this step or this step to this step, those drugs, or at least it's logical to speculate that you could use those drugs to treat or stimulate regeneration in an adult person. Okay? So with that in mind, we have been devising high throughput assays that take the cells, oh, where's the pointer? Well, that take the cells from one step to the next, and the blue arrows there show you the different steps that you could go through. So we've devised assays to go from each step to the next and trying to populate it with molecules that will drive those cells from one step to the next and eventually build, in essence, a scheme where we have molecules that control every single step. So entirely with small molecules, we could drive the process. We've built, and you can see here one of the robots that we use for screening. So, trying to get the pointer back. There we are. So here is one such assay. In this case, we've differentiated the cells in bulk up to the point where you have a progenitor here. This is a multipotent cardiac progenitor. It can give rise to muscle, endovascular cells, smooth muscle cells. What we want to know is what does it take to go from here to here. So we put those cells in multi-well dishes, as you saw on that robot, 384 well dishes, and we add molecules to individually, one at a time, to each well. And then we ask, do any of those wells turn, in this case on the image here, green, but in reality it was red. Do any of those wells turn green or red? Indicative of having new heart muscle cells being created. And in fact, we do get some hits, and this is a flow cytometry profile, and all the dots here are new cardiomyocytes that have been made, and here's a control, and there are no cardiomyocytes, and now you see them here. So doing this, we've identified a number of molecules that we are pursuing, and the list of these molecules is shown here. They target some known pathways. Growth factor, they mimic or inhibit growth factor pathways. Wnt, for instance, a molecule that's been well known to control many developmental processes, including tumors. TGF-beta inhibition, that's one I'm going to talk about. We have other molecules, and in the end I'm going to talk about a molecule that influences cardiac beating or contractility. It doesn't drive differentiation, but sustains the contractile function of the heart. So I'm not going to labor you with all of the details of what we've learned from the different molecules, but I'll use that TGF-beta inhibitor, or transforming growth factor beta inhibitor, just as an example. So what we learned from doing this is that if you look at the scheme of differentiation, the stepwise scheme going from stem cells through early progenitors, moving on to a committed cell, then making a heart muscle cell, we realize that transforming growth factor beta, a diffusible protein that's present in many tissues in the body, in particular in scar tissue, this factor will put the brakes on differentiation of this uncommitted progenitor, preventing it from becoming committed to the heart muscle fate. And the compound that we discovered that we call ITD1 for inducer of TGF-beta receptor degradation 1, that molecule, in essence, blocks transforming growth factor beta, hence stimulating cardiomyocyte formation. So what's cool about this? So what's cool about it is that we think that in the heart itself, there could be progenitors that are being blocked by TGF-beta, and that this molecule could put the brakes on the block. And so, therefore, perhaps increase the number of new muscle cells. Now, the TGF-beta in the heart is, after injury, a normal adaptive process. So after injury in the heart scars over. And the scarring is actually a problem that contributes chronically to heart disease. So this chronic increase or spreading of the scar is in part influenced by molecules like transforming growth factor beta. So what we've learned is that transforming growth factor beta is antagonistic to making new heart muscle cells. So it makes us wonder whether if we were to give this molecule, the drug that we've discovered, to injured hearts at the right time, would we basically dial down the scarring and increase then the propensity to regenerate the heart muscle. And these experiments are underway right now, and here's an example of those experiments. What we've been doing is we create a myocardial infarction, we put the drug into, in essence, a patch, a Band-Aid, and that Band-Aid, that patch, is sutured to the surface of the injured heart, and then it can deliver that molecule to the heart, and so we're looking at those animals now. We have another molecule, when delivered this way, that does offer protection. So we're testing whether the concept of whether TGF-beta inhibition can provide some amount of regeneration. Okay, so if we take a step back, what have we learned? So we used the methods of high-throughput screening to discover a heart-inducing molecule or cardiomyocyte-inducing molecule. We've identified the target of that molecule is the TGF-beta receptor. It doesn't directly block the receptor's catalytic activity, but it causes the receptor to be degraded. And that leads to the concept that getting rid of TGF-beta signaling could be a good thing, and it could, for heart regeneration, perhaps by balancing scarring or fibrosis with regeneration. And all of this, however, is not, I want to draw your attention to where this is in the drug discovery pipeline. This is not drug discovery. This is target identification, target validation. This is knowledge discovery. And this is in contrast to drug discovery, shown here. Typically, drug discovery proceeds by having a target in mind that's already understood, validated. So this is where typically pharmaceutical companies, if you were to go to Novartis or to Pfizer, they would start by nominating a target, something they believe in, they're willing to invest a lot of money in. They then develop an assay and they screen for molecules that block that target. Then they take that, they get hits, they take it through many mechanistic and chemistry studies to try to develop a drug candidate. The drug candidate goes into preclinical, and if it does well, hopefully clinical testing. And at the end of that process, which can take a decade and well over $1 billion, a drug can be introduced onto the market. Okay? But what I've told you about is target identification and validation, which feeds into this process. And I think if we question what's the role of academia in drug discovery, in the overall drug discovery process, I would argue that what we're best suited at is coming up with the molecules, the targets, understanding mechanistically how they work. Do they do what you think they should do, and they don't do other things that you would not like them to do? And can we take a process such as heart regeneration that's not traditionally thought of as being druggable and figure out how to drug it? And I think this is a good model for how academics should partner with pharmaceutical companies, because pharma is particularly well suited to the pipeline of taking that target forward. And I don't think, my view is, that academia is not particularly well suited to doing that. So I think that this offers an interest, using the tools of pharma and in engineering and medicine, I think we can identify targets and then partner with pharma to develop drugs. So how do you identify new targets that you'd like to take to Novartis? So it's a bit, it's a needle in a haystack. A cell is so horrendously complicated. We all know that there are about 20,000, 25,000 genes. Okay? What's not commonly thought of is that there are 1 million proteins from those genes, if you think of all of the variants and modifications and so on. The entire pharmacopoeia that we have available to us just scratches the surface, right? It targets only several hundred different protein targets. I know drugs are not all selective for particular targets. Many of them target multiple things. But generally, they're thought to target or designed to target only in the hundreds. Okay? Typically, even if you look in those hundreds in depth, they fall into a few classes. G protein coupled receptors, kinases, nuclear hormone receptors, molecules, ion channels, things we understand fairly well. So an open question is, how many druggable targets are there out of the 1 million proteins? I'm certain that the vast majority are not good drug targets. I'm not advocating that. But I think we need to sift through all those other proteins. And that's where the high throughput biology comes in. So the chemical biology and functional genomics enterprise is basically to take libraries of molecules and you dump them on assays that have some complex biological assay, such as stem cell differentiation to cardiomyocytes. And at the end of the day, through deducing the targets of the molecules you dumped on, you can generate a network. And that network controls the complex biological process. And if you understand that network, I think you would be able to go in there and identify what would be the really good drug target, right? Now, if you think about systems level understanding of something and networks, I mean, it's sort of obvious. If you look at London from the air, and you're thinking about just going from one part of the city to the other, it's horrifically complicated and you can't possibly imagine how to get to there from here. Okay? On the other hand, if you fall into the subway and you look at a train, it doesn't do you any good either. It's too detailed, too high resolution, and it's a single point, single node, one subway station. So what you really need, as you all know, is a tube map. And if you look at the tube map, you can find that there are really important nodes, where everything comes together. And if you muck up those, you're certainly going to change the process. On the other hand, there are nodes out here, who the hell lives in Tufnell Park? So, if you want to influence the train that's over here, actually, if you want to influence the train going north, that's the station you would block. If you want to influence the whole network, embankment. So the point is that from understanding the network, you can identify the drug targets. So the assay I told you about, where we put the stem cells in the dish, we add libraries of molecules, and you end up with heart muscle cells, hopefully can give us that network. But what we learned is that it's not so easy. The example I told you about, I screened a drug discovery library. Okay? And there are many different types of chemical libraries. So the library I screened was a large diversity collection, basically suited to identifying drugs. By their very nature, medicinal chemists, or not the nature of the medicinal chemists, by the nature of the molecules in the library, they're promiscuous. The medicinal chemists, I don't know if they're promiscuous or not. But the medicinal chemists dial in features into molecules that exist in these large diversity collections. So these molecules have to be orally bioavailable. They want them a certain size. They want them, because chemical space in the drug, if you dial in those criteria, it's 10 to the 60 compounds. You can't screen 10 to the 60 of anything, no matter who you are. So what they do is they create diversity sets where they have representative molecules that are promiscuous. They're floppy. They can interact with multiple proteins. So if you screen these libraries and you come up with a molecule like we did, it takes 7 years to figure out that it interacts with TGF-beta for one molecule. That is not a system. That's not systems level understanding. So what you need is, if the assay is a black box assay where it doesn't have the target, it should be screened against a library where the components of the library bring the target information. And that you can do by screening focused libraries, known drugs, or libraries of molecules that inhibit particular proteins in a network. You can screen little RNA molecules that go in and block every single gene in the genome, called siRNAs. These go into the genome. We have siRNA libraries where you can screen individually, in essence, an siRNA for every single gene in your body, and and you can then ask which gene controls, say, stem cell differentiation to heart muscle. MicroRNAs are a natural version of those little siRNAs, but they have a built-in degeneracy, whereas the siRNAs are specific, one siRNA, hopefully, one gene. MicroRNAs, one microRNA, 300 genes. And although it's not trivial to find the target, you can, and I'll get back to that in the next couple slides. So the focused libraries allow you to decipher the network controlling a complex process. So to put that in very schematic terms, what I've told you is that if you have an assay, like you know the target, some enzyme, and you want to block it, and like let's say you're at Novartis and you have an enzyme, you want to block it to cure some metabolic problem or treat a metabolic problem, you should be screening a large unfocused library and look for your drug. But if you have an assay that doesn't, where it could be, you don't know what the target is, you screen to identify the target, you shouldn't be screening it against an unfocused library that doesn't bring the target to the table, but rather a library where you can go from a hit to target. Okay, so I happen to like microRNAs. And the reason is that there are relatively few of them, maybe only 1,500 in our genome. Bob can probably correct me and even tell me the precise number. But there aren't all that many. And these microRNAs work by suppressing multiple proteins. And through knowledge of those proteins, you can come up with a network. And you can then hopefully construct that network and figure out where the good drug targets might be. So I'm going to give you an example of using, in the last example, will be an example of using microRNA screening to understand heart muscle contractility. So if you think about this overall developmental progression, as I said, the last step is particularly challenging. How you go from an immature myocyte to one that beats nicely, robustly. No, it's very hard to think about how to perform such a screen, much less screen for something that would affect contractility in the adult heart and possibly could be used to sustain contractility in the face of heart failure, which causes basically a decline in the contractile function of the heart. So to do this, I had the good fortune of teaming up with an engineering colleague of mine, Jeff Price. And Jeff and his colleagues were experimenting early on in those days with optical methods of recording from excitable cells like heart muscle cells. So what we teamed up to do was to build an instrument called the Kinetic imaging cytometer. Jeff coined this term. And basically, this is a microscope that takes pictures of excitable cells in high throughput, one well after another. And what you're looking at here is a movie of the calcium transients, the calcium release inside a heart muscle cell derived from these human-induced pluripotent stem cells. And these calcium transients, they basically are firing every time the heart muscle cell is contracting. So the instrument takes a movie of this, but then the instrument will segment that image into images or movies of individual cells, each cell in that well. And here's actually the commercial version of this instrument that's just been built now. So the value of this, of single cell imaging, is that the previous movies were beautifully synchronous. But here's what happens if you dump a drug on that is arrhythmogenic, right? It causes some sort of asynchrony, some arrhythmia. Actually, this is a drug that's arrhythmogenic in people. And so you can see the effect that this drug is having on these cardiomyocytes. And so they're firing at different times, okay? And so if you look at these, if you were to record from the whole well they'd cancel each other out. And you can see that just looking at 3 separate cells here. You can see the green trace canceling out the red trace. And so if you look at the whole well, in essence, it flatlines. But if you look at individual cells, you can make out the individual kinetics. So this instrument is very useful for predicting the arrhythmogenic effect of drugs, at least the cell autonomous, the drugs that would be arrhythmogenic for cell autonomous reasons. So that's a major effort in the lab and at Jeff's company, Vala Sciences. But I'm enamored for this machine because of its ability to look for molecules that will control the last step in stem cell differentiation or cardiac contractility. So here in one slide just diagrams the overall approach we've taken to developing an RNA-based drug, which I think is very well suited to this problem, an RNA-based drug to treat heart failure. So here, we basically build an assay. We take a target. This is a protein that is important in maintaining cardiac contractility and in both the normal and in the failing heart. And we look for microRNAs that are going to suppress that protein, right? The idea being that in a failing heart, if that microRNA is there, that we could come in later with an anti-microRNA and block its function. We could develop an antimer. Okay? So we do a screen. Here's a positive and negative control. You can see the green when we have not blocked it, and we get rid of the green with a particular positive control RNA molecule that's specific for the target. Now, if we screen the entire 900 microRNAs that we have from the human genome, what you see, and you array them in a type of a plot that we call a volcano plot, but you array them, all the ones in yellow and orange here are the hits that we like. They all are capable of suppressing the particular target. So we then use the kinetic imaging cytometer to ask, okay, they suppress the target, do they also suppress the contractile function of human cardiomyocytes in a dish? And so contractile function is scored in this y-axis here. The higher up, the lower down you go, the more contractile it is, the faster it is. And so what we see here is that a negative control microRNA is at this level. This is a distribution of all the different hundreds of cells that we looked at. This is the average. This is the one standard deviation. This is a 95th percentile up here. So you can see this distribution. A positive control to the particular target slows it down, and the microRNA is really good at suppressing contractility. So then we come in with an anti-microRNA to block the microRNA, and you can restore it. Okay? So you can restore the contractility deficit caused by the microRNA. So then we said, well, what happens if we do this in an animal? This was, of course, in vitro on the kinetic imaging cytometer. But here we take an animal. This is an animal where surgically we've gone in and created chronic hypertension. We've ligated the aorta. Now you've put constant pressure on the left ventricle of the heart. Over a period of months, that heart will begin to become hypertrophic, to dilate, to first enlarge, eventually to dilate. At 3 months after the ligation, that heart is dilated. And if you measure heart function, here's one measure of heart function. You can see the control animals, they pretty much hold steady. It's a mock surgery. But the animals in red here are the animals that have basically received the control anti-microRNA, and with every month they get worse and worse and worse. In fact, many of these animals out at this point are dying, so we're looking at the ones that are actually surviving here. Now, at this time, 3 months, we started infusing the anti-microRNA or the control microRNA. And in green are the animals that received the specific anti-microRNA. And you can see we halted this progression of heart failure. And in fact, there was even a noticeable restoration of cardiac function. And this extended not just to the cardiac function. Well, there was another parameter of cardiac function, hemodynamic one shown below, but I won't go back to it. But it also reduced the scarring that occurs that I had mentioned before, and this enlargement of the heart as well. And you can see the difference here. If you look at blue, which is the scar tissue, here's an unaffected animal. Here's what happens in the heart failure animal that has received a negative control, and here's the anti-mirror, which reduces the blue, and that's quantified here. More blue, less blue. And here is the size of the individual muscle cells as well. So what I've told you then, in this last vignette, is our attempts to use screening to develop an RNA therapeutic for failure. But I think the real strength of this is that we didn't just get the one molecule I showed you. As you remember all those yellow and orange dots on the screening profile, we got many microRNAs. And so what we're trying to do is identify the targets of each of those microRNAs and construct the network that controls cardiac contractility. Of course, a lot's known about cardiac contractility, but what we're hoping is that we will identify through this process really good druggable targets that can control contractility, and then something that if I phoned up Novartis and I said, look, I've got a target that controls this, they would answer my phone call. So overall conclusions then from what I've told you this evening, that drug target space is much larger, that drug target space is potentially vast. The current available pharmacopoeia only scratches the surface, and there are probably many more proteins that we need to, certainly in this post-genomic, post-proteomic era, sift through and identify good drug targets for diseases that weren't considered druggable, like heart regeneration, or problems that aren't considered druggable, like heart regeneration. And I think the answer to that is a high throughput biology, chemical biology, which is screening with molecules, synthetic molecules, or functional genomics, which is screening with RNAs or proteins like microRNAs, siRNAs, or even libraries of known proteins. And finally, I think this gives us an idea of or I would propose that this makes for a good relationship between pharma and academia. That academia is particularly well suited to this target identification, drugs, chemical biology, functional genomics enterprise, and that once we have the fruits of that, those networks, that's where we can begin to have this good handoff to pharma. We could validate the targets, perhaps by identifying tool compounds, maybe not something that pharma would want to take as a drug, but molecules that would be useful for animal studies, validate the target, and then pass that on to pharma or to a biotech company. So I'll stop there. Thank you very much. Sure.
Floor is open for questions.
When you say that you understand the network for this processor, this is a loaded statement. These connections are non-linear and you don't know them. So how much approximation or how much post-scaling can be tolerated?
How much, say that again, how much what?
How much approximation?
That's a great question. Okay, so here's the problem. As you point out, you build a network. The network is very fuzzy, right? So I have this discussion all the time with the systems engineers that we collaborate with. They're used to looking at data sets that others have generated, and then they could do statistical analysis and come up with what they think of as a high quality network. I don't want this high quality network. I want the nasty, fuzzy network, because we can go back and pick siRNAs to every one of those nodes and test them individually. Thousands. I can test tens of thousands. So nasty, ugly networks where we don't really know the arrows are great. So, how these networks are built at a systems level is that the systems biologists, they look at all your proteins and they link them together based on the literature. One protein is known to interact with another, but that might be in some kidney cell, not in someone's heart cell. So I don't care. Even I would want it to have all the false positives. I just don't want the false negatives. Yeah, that's a very important question. Back there's one.
What's the method of delivery for the
Jeff.
if you have something that's going to affect lots of means is there a way to target?
Yeah, that's another good question. How do you deliver an RNA therapeutic is basically what you're asking, right? Yeah. So that's an important question. I glossed over that. So what we've done is that we formulated this in a lipid vesicle and injected it intravenously. This is a straightforward kit you buy from Invitrogen, so we did nothing special. This is in essence has been used a number of times, in particular by people at Miragen, a company founded by Eric Olson and his collaborators. So as it turns out, the heart is really good at taking up these lipid vesicle encapsulated antagomeres. I understand skeletal muscle, damaged skeletal muscle is also fairly good, and of course the liver. But I think from people at Regulus, I understand that you can even put in naked antimeres if they're the chemically modified type, and they will go to certain tissues. But so the tissue tropism depends upon how you formulate it. So they don't go everywhere. And I think, but since we're targeting the heart, that is actually an organ that takes up these antimeres very well. I can't speak to other organs. I imagine it doesn't get taken up very well by many organs. One last thing is we did select this particular microRNA for follow-up because it's not expressed in very many other organs in the body. And so the antimere, even if it were to get taken up by the liver, is not going to have, or at least I don't think it would have any effect. So we chose a test case where it was relatively easy. Oh, sorry.
You actually have receptors to the vesicles and
No, we didn't. There are much more sophisticated ways to target. This was just using this Invitrogen, I think they call it in vivo delivery reagent, something like that, where it's just basically a lipid vesicle and it's shot into the bloodstream. Yeah.
The cell differentiation, you get hundreds, thousands of cell types, whatever. So how do you deal with cell autonomous versus non-autonomous effects of the library that you're putting on?
Yeah, that's another really good question. Okay, the question is, in our screens, we have heterogeneous cultures. How can we tell that the molecule we're adding is acting on the cardiomyocyte progenitor or on some other cell in the screen? We can't, a priori. And so we have to, in subsequent studies, work that out in terms of its mechanism of action. I would love to be able to put always pure cells in a dish, but that's often technically not feasible. In the case of the molecule I was telling you about, we do know it's directly on the progenitors. I have other molecules where we do know it's on other cells in the dish, and then it causes them to produce something. So that becomes a developmental biology type problem.
Because of the time, I think we'd like to stop now. If you want to come up and talk to Mark, he'll be here for a few more minutes. Let's thank him for a great talk.
Thank you.